My name is Josefina Campos. I'm the director of the National Center of Genomics and Bioinformatics, Malbrán Institute, in Argentina. We are the national reference lab for the Ministry of Health. So our main objective is to monitor pathogens of public health relevance, among other non-transmissible diseases. I have been working in monitoring these pathogens for the past 15 years. One of the things we have to have in mind from the National Reference Lab is how the surveillance system is set up. What do we have in place and what do we need? With that, we have to have in mind the geography, which other reference lab we have across the country, and how we want to monitor the dynamic of that particular disease. When we need to think of new technologies, like how genomics is merged in this surveillance system, this is one of the questions we need to ask. For example, in Argentina, all the National Reference Lab for all the pathogens of public health importance has very well-established laboratory surveillance network. So how a new technology can kick off? One of the things we can think is, where are the gaps in this surveillance and how we can fill those gaps? We have examples, for example, when we study outbreaks where the technology we have in place was not enough to discriminate if the isolates were part of that outbreak. For example, cases of Streptococcus pyogenes in 2007 in Argentina. But we also want to do a continued monitoring of some other pathogens. For example, those of foodborne associated outbreaks. In that case, we want to know what is circulating in the country and how this can be linked through the best technology we can have. In this case, genomics tend to be essential. That's how we start working with PulseNet Latin America and the Caribbean, not only in Argentina, but across the region, to do the surveillance of foodborne and waterborne pathogens. We have many other examples, like, for example, the Neisseria gonorrhoeae outbreak, where we really need genomic surveillance to set up and identify 2 different clusters in the country in different provinces. This has a direct impact in some policies that the Ministry of Health eventually take to prevent new outbreaks to come. One of our major outcomes in 2020 was to use the genomic infrastructure we set up in our institute to work to determine the resistance for TB isolates with the National Reference Lab. This was because we worked before in a specific project making a case study for this. We also have a very well-known example of Vibrio cholera, that when cholera kicked off in Argentina in the 90s, we could identify through genomics different lineages, the pandemic and the non-pandemic. Even now we have pandemic cholera-free in Argentina, we still monitor every cholera suspected case with genomics to know if this is a pandemic lineage or not. Why do we do that, and why we don't use the traditional techniques? Because even with the traditional techniques, we can get up to Vibrio cholera, and also we can get to know if it's toxigenic. We don't know if it's a pandemic lineage or not, and that has a direct impact in the response in public health. And we work with that with every national reference lab in the institute to try to understand how new technologies can actually merge. Of course, it was essential during the COVID-19 pandemic, where we established a completely new genomic response for the whole country. And that, of course, because of the geography of the country and the amount of samples that we were receiving at the National Reference Lab, this was impossible to sustain. First of all, in our unit, we have scaled up our genomic capacity. How? Getting new sequencers and doing automatized robots for the library prep, for example. But even with that was not enough for the genomics response. So we set up a new genomic network across the country where all the provinces has now all genomic capacity. But this is also a challenge, not only for creating the capacity across the country, but also to link this capacity with the existing laboratory surveillance network well established. We're still working in which pathogens these new technology are actually making a difference and which ones have to be done locally, centrally, or is not needed at all. This all has to do in how the public health response is think for each particular pathogen of importance.